I'm David Mail from Department of Life, Health and Chemical Sciences, and some of the work that we are doing is involved in trying to work out how to get therapeutic agents into the brain. And this is a really big problem for the pharmaceutical industry because 95% of the drugs that we would use to treat diseases like multiple sclerosis and Alzheimer's disease don't actually get into the brain because they don't cross the cells lining the brain blood vessels, which are called endothelium. And as far as gene therapy is concerned, it's even worse because none of the potential genes that we might use ever get into the brain. Well, we work with a number of different companies in different ways. On this particular project that we've got going on here, we've been collaborating with an industrial partner called Midatech, who are interested in how nanoparticles can be used as carriers to get drugs into the central nervous system. And this particular project involves a variety of tissue culture techniques in which we apply the nanoparticles to the cells that we're using. We've developed already a variety of different sophisticated three-dimensional cultures of the human central nervous system. And then once we've got the nanoparticles onto the cells, we'll take them downstairs to our electron microscope unit and see where the nanoparticles have localized in the cultures. We have a whole variety of excellent facilities here, including the tissue culture, molecular biology, transmission and scanning electron microscopy, fluorescence microscopes, FACS analysis, and so on. And we apply whatever are the appropriate techniques for the particular problem that we are presented with by industry. In this particular project, we're working with a company called Midatech, who produce gold nanoparticles. But for some of our other projects, we're working with a company called MedImmune, and they are interested in the development of aptamers for targeting cells in the central nervous system. A lot of the work we do uses fluorescence microscopy, both on cells in vitro and on tissue from experimental animals or patients. Some of the studies require time-lapse video microscopy, but in the nanoparticle project we have been using primarily electron microscopy. We've been using transmission electron microscopy to identify exactly how the nanoparticles cross the endothelial cells, and you can see some of the results that we're getting on the screen here. So for example, this is an endothelial cell, brain endothelium, and the nanoparticles are identified by silver staining, and in this particular section what we can see is the nanoparticles crossing the cell, either going through the cytoplasm here or in some cases by the vesicles and emerging on the brain side of the endothelial cell. And we think that this will be extremely useful as a mechanism for getting drugs and therapeutic agents across into the central nervous system. Now, a lot of this work has been done in association with Heather Davis here, who is manager of the electron microscopy suite here, and it's been using one of our microscopes here. So tell us a little bit about it, Heather.
So this is a high contrast transmission electron microscope in the suite here, and we also have a 200 kV high resolution transmission electron microscope and a scanning electron microscope, which is also high resolution.
In this particular project we were working with our industrial partner Midatech, and the work so far has generated one publication and also one joint patent application, and we are continuing to develop the work. It's been funded in a variety of ways. They sometimes will support particular projects for maybe 1 or 2 months, or in this case supported a research assistant for 9 months to work on this particular aspect. And in other cases we have joint funding of PhD students. There are a whole variety of different ways that we can work with industry, but we work very effectively because we share our data and we discuss carefully the best ways forward at each stage. Most recently, Midatech wanted to analyse the quality of material from their manufacturing plant, And in conjunction with them, we decided that scanning electron microscopy was the best way to check for the uniformity and the quality of the materials that they were working with. And we are now doing this work as a short contract using scanning electron microscopy. Initially, we did a pilot study comparing scanning and transmission electron microscopy, and we decided at that stage, in collaboration with a company, that scanning EM was the most effective. And you can see some of the results on the screen here, where we've got the strips that they wanted us to analyze, and we're starting to work on image analysis of them to see how good they are and how uniform they are. We've developed excellent relationships with the companies that we've worked with, mostly because we discuss carefully what is the best and most effective and also the most economical way of finding out what they want to know.